We proceed to a more interesting topic, cerebellum. So, cerebellum is located somewhere over here. I have explained in anatomy again how to recognize that in image, how it looks like. You have two lobes and you have the vermis in the center, vermis. So, two lobes, vermis, lobes are connected to the vermis. So, the central part of it is the vermis, and then you have two, and the convolutions on the surface of the cerebellum are called folia. There are big fissures which split it into multiple parts visually, but functionally you have a different kind of arrangement. So, these are certain things which you need to be aware. So, I will go back to the pictures. So, that is the cerebellum, cut section of the brain. So, this whole part is the cerebellum, is included, cut section. So, you have this leaf like this one, each of which is called as a folia. Now, this is the midbrain, pons, medulla, spinal cord, occipital lobe, and to give you an understanding, they have got the fourth ventricle over here, and there are things which connect the cerebellum into the spinal cord and to the brain. So, they are called as the peduncles. Where are peduncles? Peduncles is not written over here. If you look at it from the this is from the back, from the surface of the brain, you see it like this. There is something called as the primary fissure, which is over here. Structurally, we look at it in a different fashion. I think I should explain a little more. You have the cerebellar cortex, and then you have the deep nuclei. The input and output is through peduncles. Superior, middle, inferior. So, up, middle and lower down. So, that is technical stuff which you would the cortex as such has multiple layers, granular, then you have the Purkinje cell and the outer cortex. We will look into this a little more detail, lot more detail actually. So, there are so outside it's there are two lobes, central vermis, then within each lobe you have the cortex, which is the which cortex which contains multiple other layers. Now, these are microscopic, you see under the microscope. So, you would find these layers. I think this is wrong, I will correct it in the picture. So, 3 layers. Then you got the deep nuclei, which is sort of replicates the in the anatomy of the cerebrum, in which you got cortex and basal ganglia. Only here it is called deep nuclei. You have got dentate, emboliform, fastigious, and globose, I think DFG. So, that is how you remember. These are just for mentioning, not something which you need to remember, because they are structural names. So, they do not actually correlate with function. So, functionally you have floccular nodular, everything in anatomy is a mouthful. So, that is vestibulocerebellum, then spinocerebellum, and I think I should take the help of the diagram, and cerebrocerebellum. Flocculonodular node is part of the lower part of the vermis and the little bit of the deep gray. The spinocerebellum is the intermediate part, and everything lateral is the cerebrocerebellum. Basically indicates that it is connected to the cerebrum, the cerebral cortex, and there is a lot of communication which happens, and obviously the larger areas are is connected to the larger part of the cortex. A brief description: vestibular cerebellum is basically with the vestibular apparatus. So, that would be in position. Spinocerebellum is with posture and tone. This is for movement control. Control can be anything, anything, hand, speech, leg, all those things, movements of various kinds are controlled by the cerebellum, and we have been able to demarcate these locations to be responsible for these actions. So, there are descriptions of again topological organization within the cerebellum, but they are not in conventional teaching. So, again not very well defined as you would see in the sensory homunculus or in the motor homunculus, but there are maps within the cerebellum. So, these are the central part of the cerebellum is the older part, in the sense that these are there in lower animals also. This is higher, so newer. Higher animals, in the sense that you have a higher amount of cerebral cortex where this connection is relevant. So, that is about the structure of the cerebellum. What is more interesting is this three layers of the cerebellum, and that is something which I have to show and explain to you. So, we start cerebellar circuitry. So, this is about cerebellum. My first impression of it is it is a monotonous circuit. You see a section of it, it gets replicated all across the cerebellum. So, what I mean to say is that what cerebellum does for the speech would be exactly the same stuff it does for movements. So, the kind of function which is the servo mechanism which is performed by the cerebellum remains the same across various kinds of tasks, irrespective of the nature of the task. So, speech is regulated in the cerebellum, movement of the hand limbs is also regulated in the cerebellum in almost same fashion by the same circuits which I am about to show you. I will be going back and forth, so please do bear with me on that. I spoke about three layers in the cerebellum. So, the major cell in the cerebellum is the Purkinje cell. So, it is a pretty large cell, it has got a dedicated layer for itself. It is a large cell, it has got multitudes of branches. So, very large number of branches, and the Purkinje cell is like a tree. The Purkinje cell synapses on a single deep nuclei. So, the deep nuclei is the output. So, Purkinje deep nuclei, and that is the output. So, Purkinje cell basically is inhibitory over here. Now, there are two kinds of inputs. So, we have discussed about the output. So, what about the inputs? Now, inputs into the cerebellum come from two sources. One of them is called as the inferior olivary nucleus, which is in the brainstem, and then you have got fibers which there are two kinds of fibers. So, they are in turn called mossy fibers. Anatomical names are difficult, peculiar, and I have difficulty remembering this stuff. I do understand the concept, but please do forgive me if I am making mistakes in terminologies and naming. Now, the fibers which come from the inferior olivary nucleus are called climbing fibers. Okay, so there are two sets of these fibers. So, we will remove the inferior olivary nucleus for that purpose. We will just keep it simple and quote climbing and mossy fibers. Now, climbing fibers are interesting because they come from the inferior olivary nucleus, and these fibers I need different color. One single fiber would go and branch multiple times synapsing on a single Purkinje cell. Okay. So, the inferior, it also sends another this one to here to the deep nuclei, and that is positive. So, you got a negative there and a positive there. Now, mossy fibers on the other hand, I need colors. Mossy fibers go into the granular layer, and then they synapse into something called as a granular cell. So, once the granular cell is like this, no, I need the granular cell has an interesting method. So, it sends fiber up, goes all the way over here and synapses with multiple Purkinje cells. So, one signal is distributed 1, okay, next I think. If we look at it from the Purkinje perspective, one to one for climbing fiber, many to 1 for a mossy fiber. 1 to 1 is climbing fiber. So, climbing fiber, one fiber goes to one Purkinje cell, whereas mossy fiber goes to a granule cell, and many granule cells would synapse on one granule cell would synapse on multiple Purkinje cells. Conversely, one Purkinje cell gets information from multiple granule cells. So, the net output is negative. So, that sort of, so this kind of mechanism in which you get information from two sources, one goes specifically to a cell, makes it reduces the input, the other one spreads, there is a local spread in that region, because wherever this fiber goes on, it gives a little bit of stimulation to the Purkinje cell. So, there are two sources which converge onto the Purkinje cell. It again it averages this signal. So, average is spatial averaging for the mossy fiber, and point, I do not know how to term it. So, you have point stimulation from the climbing fiber. Essentially, it compares two sets of information and generates an output. The output of the cerebellum in turn goes into the deep nuclei, wherein it gets information from other sources also. So, you have two positives and one negative, and the sum of all of these things is the output. Describing this network is a very idiotic process. Ideally, somebody should model the whole thing, but there are models of computational neurobiology of Purkinje cells from quite some time, but apparently the modeling is very complex. I would invite people in the audience to model it in a simpler fashion, taking some assumptions. I am pretty sure that there is a Nobel moment in the cerebellar network which is just there for the taking. So, that's how I would like to show. So, the diagram, I come back to the diagram. So, this is the climbing fiber which I was telling, coming from all the way from the inferior olivary nucleus. Remember, there are two sets of output from the inferior olivary nucleus within the cerebellum. Sorry, it is input. So, this is an input. So, climbing fiber goes to one Purkinje cell. So, this is one nucleus, one Purkinje cell, and then it modulates that activity, reduces the output of the cerebellar deep cerebellar neurons, whereas this is positive. So, one single neuron generates a positive and a negative response on the output nucleus. Now, if you look at the other fibers, this is another set of information, comes to the granule cell, then goes to multiple parallel fibers. So, multiple parallel fibers go across to multiple Purkinje cells, and then that produces a change in the negative stimulation, where again this is positive and this is negative. So, there are two sets of data which go into the cerebellum. one from the mossy fiber and the other from the climbing fiber. The information is sort of compared, and then you have an output which is negative, compounded with outputs inputs from the input nuclei into the output nuclei. I think it makes it more confusing, but yeah, what is important to note here is these two loops which are there. So, these two loops are responsible for a lot of this for the interesting mechanism which the cerebellum does. Now, what happens if the cerebellum is not functioning? So, reaching a particular object, say this object is in free space, you can execute the task, but there is a tendency of overshooting. So, if I were to say a standard test in cerebellum, testing cerebellar function is do this finger object test. So, instead of this there would be the examiner's finger, and the person has to do this fast. I am able to do this fast, but in a patient with cerebellar dysfunction it would do this, you go back, touch somewhere else, go near, but overshoot, either you overshoot or undershoot to target. So, conversely the cerebellum ensures that reaching a target is, it accurately does it for all of us. So, this particular network which I have shown here is responsible for the job. Now, apparently you can develop a learning model also. Learning model, it is not developed, it is there within you. So, how learning happens in the cerebellum is somewhat like this. So, you have an air puff and a tone, you respond to So, this is conditioned reflex. It's, you must have heard of Pavlov's reflex. So, inferior olivary goes into the Purkinje and that reduces output over here. Mossy fiber, which is from the ear, goes over here, stimulates here, again negative here, and then changes the output over here. So, after a certain level of stimulation through these pathways, the gain of the system of this system changes. And that is learning in the cerebellum. So, you repeat a task, these parameters change in the output nuclei due to these various networks, and you achieve a steady state. So, that steady state is reflective of the maps which I was telling you. So, this is a task map. So, a task map being represented in terms of synapse values. So, the more the synapse gets stimulated, the more neurotransmitters are released, and then it achieves a new equilibrium. So, a particular signal generates a particular amount of neurotransmitters, and synaptically as the signal increases, the synapse output increases until you reach a new steady state. So, the specific state function of this set of synapses would constitute a mechanism of learning, and the association is here. So, you associate one stimulus to another stimulus, and then that causes a steady state change in this output nuclei, which is the learned response. So, that is how one of the mechanisms of learning and how cerebellum contributes to learning. I have also discussed how tasks are smoothened. So, the smoothing happens in two mechanisms, one all of that in the Purkinje, this one through this single input high dense signal, and the other one is through this sparse signal. So, this is a dense signal, this particular thing is a dense signal, and this is a sparse signal. Sparse because multiple granule cells impinge, give a small signal to the Purkinje cell. So, the Purkinje cell averages both of this. So, the dense signal and the sparse signal, and then outputs it into the deep nucleus, which in turn gives the final output of the cerebellar computation. So, please do feel free to read about this topic. I do not know if I made justice of it, because what is written in medical textbooks is just about like this. Unlike many other topics which I have dealt with, I did not know how to simplify this any further than saying point A, point B and stuff like that. But as I have highlighted in the section on vision and some other sections, this is a very important network. Actually, I feel it is very easily codable also, but somebody should actually look into it. The outputs which you would get from coding a network like this would be really very interesting. And I think I take leave at this place, completing the topic on movement, the pathways involved, the mechanisms which we understand as of now, and a brief description of the cerebellar function which I have highlighted over here. See, cerebellar function is the key point in the whole thing. All of it is how of it is, not the how of it, how the path exists and which part of the brain or the nervous system is involved in coding for individual components. What is shown in the cerebellar network is a very interesting mechanism. I have not found an engineering equivalent in which data of this kind, sparse and dense, and you got an averager, and then you blend all together in the deep nucleus. So, I have no idea of anything which resembles the cerebellar pathway, but its importance is highlighted by the fact that it is a very monotonous system. So, you got the entire cerebellar cortex looking exactly like this, repeats, but it does anything from learning music to driving cars to speaking properly. Thank you.